Okay, so we're going to start off with our assignment here. Indicate whether each of the following statements is true or false. If false, explain why. A DNA triplet code is made up of 3 amino acids. This is false. It's not made up of 3 amino acids. It's made of 3 a triplet code, a nitrogen base, and that tells us the amino acid to make. So it's made up of 3 bases, 3 nucleotides. Right? DNA triplet code for the same amino acid as another triplet code. Yes, true. Adenine bases can only bond to cytosine bases. G can't. Adenine bonds to, oh, no, not cytosine, this is thymine. So this is false. A double helix is similar to a spiral staircase. Sure. Genes provide instructions to make proteins. Yep. There are 10 different amino acids. I think there's 20. Histone is one of the 4 base pairs. False. A histone is what DNA wraps around. Alternating phosphate and deoxyribose sugar make up the backbone of the DNA with the base pairs attached in the middle. Yeah, that's good. During replication, the DNA breaks into smaller pieces and reforms into 2 smaller halves. So during replication, the DNA breaks into smaller pieces and reforms as 2 smaller halves. I don't know. It's not really smaller halves. It just into 2 DNA strands. It breaks in half and then makes 2 DNA strands. I don't know. False. Hard to tell with that one. A DNA molecule has 3 strands of nucleotides braided together. No, it's just 2. Okay. Use the DNA triplet code with their corresponding ones. So I'm going to look it up here. So first one is valine. So just use the first one if they don't give it to you. GTT. Alanine. GCT. And the asparagus one, AAT. Okay. And then find the complementary base pairs to that. So the complementary base pairs, remember GCAT. So G's go with C's, T's go with A's. Like that. Right? These 2 go together, and these 2 go together. Find the complement. Oh, that was what we just did. And that's all. So let's do some sample diploma questions. Okay, so gene segments may initiate, terminate code for amino acid sequence. The number of amino acid gene segments shown for the code above. So we'll look up ACG. And if you look up ACG, it is threonine. And you look up TAG. Where's TAG? TAG. Oh, it's stop. So if we're looking for the number of amino acids, there's actually 1 amino acid here. Got to be careful with that. Okay, match each of them. So with the GCAT, cytosine goes with guanine, and thymine goes with adenine, like that. And then directly attached to the nitrogen base is the sugar, and then the phosphate is what attaches the different nucleotides together. Okay, so phosphate is 1, sugar is 2, guanine is 3, and adenine is 4. In humans, the WRN gene on chromosome 8 is responsible for unwinding and separating 2 strands of DNA during DNA replication. 2 components of DNA that would be responsible for separating. So the thing that separates is during DNA replication. If we look at this diagram right here, it splits right down the middle. So it's the 2 nitrogen bases that rip apart. So adenine and thymine, or 2 nitrogen bases. They were the ones that break apart. Okay, and let's do this lesson on mutations and genetic diseases. So curriculum, describe how mutations in DNA affect proteins. They can result in human diseases. Describe the development of resistance to bacteria and viruses based on concepts of mutation, plasmid transfer, and transformation and natural selection. So make sure you know what these 3 terms are, because they're in the curriculum, so I will test you on them. Okay, we need to know how pedigree charts work, and do trends with autosomal sex linked, which we kind of did, but we'll talk about it with genetic diseases this time. Okay, so a mutation is a change in the sequence of bases along the DNA. So most occur naturally, or they can be from radiation exposure, chemicals, carcinogens, things that cause cancer. That can change your DNA. So each change is carried to the next generation and inherited by the offspring. And mutations are good. They increase variability and lead to adaptations. So some of them are beneficial, some of them are harmful, right? And when everyone thinks about mutations, they think about, oh, a mutant, like Cyclops shooting lasers out of his eyes. It's not really realistic, because he'd have to eat like so many calories a day to account for that laser. The energy doesn't come from nowhere. But most likely what mutations we actually see are different colored carrots, right? So it might be beneficial, maybe it tastes better, maybe more rabbits eat it and then they poop out seeds and spread the seeds everywhere. Who knows? So there's a couple mechanisms, we're not there yet. So mutagens are agents that are responsible for mutations. Mutations are like typos in DNA, right? Normally, if I spell something wrong, you can still read it, right? Nothing is spelled there other than the 3-letter words. So most mutations are the same way. So you get little mutations, they have little effect on the product produced, but yeah, sometimes they're worse than that and they can be bad. Mutations usually occur in chromosomal regions where they do not affect the protein, and body can repair some of the mutations. But some mutation cause genes to stop working, they alter their function, and those are the really dangerous ones that would be bad. Okay, so here's a video on mutations and natural selection, and if it doesn't work, it's because YouTube blocked it.
This episode of Stated Clearly was only possible with support from our viewers and from Brain Tools LLC, a company dedicated to developing and delivering to patients new treatments for Alzheimer's. Stated Clearly presents, what is natural selection? Natural selection is one of several key concepts contained within the theory of evolution. To understand exactly what natural selection is and why it's so important, let's first take a quick look at 2 other evolutionary concepts, descent with modification and the overarching idea of common descent. Descent with modification is the observable fact that when parents have children, those children often look and behave slightly different than their parents and slightly different than each other. They descend from their parents with modifications. The differences found in offspring are partially due to random genetic mutations. Common descent is the idea that all living things on Earth are related. They descended from a common ancestor. Through the gradual process of descent with modification over many, many generations, a single original species is thought to have given rise to all the life we see today. The common descent of all life on Earth is not a directly observable fact. We have no way of going back in time to watch it happen. Instead, common descent is a conclusion based on a massive collection of facts. Facts found independently in the study of fossils, genetics, comparative anatomy, mathematics, biochemistry, and species distribution. Because the evidence for common descent is so overwhelming, the concept has been around since ancient times. In the past, however, it was rejected by many philosophers and scientists for one main reason. You cannot get order and complexity from random chaos alone. The bodies and behaviors of living things are extremely complex and orderly. Descent with modification simply produces random variation. All through history, no one could explain how complex life arose from simple life through random variation until Charles Darwin discovered natural selection. Charles Darwin, who lived from 1809 to 1882, was a naturalist, someone who studies nature. At the start of his career, he traveled the world by ship, collecting and documenting plants and animals. During his travels, Darwin became very interested in the idea of common descent. He noticed that islands contain species of plants and animals unique to those islands. They can't be found anyplace else on Earth, but they often look and behave surprisingly similar to creatures found on nearby continents. Tortoises on the Galapagos Islands can be distinguished from those of Africa. Meanwhile, with the exception of size, they're almost identical to a species found nearby in South America. Darwin believed these similarities could be best explained through common descent. Long ago, a tortoise from the mainland may have drifted to the islands, possibly on a raft of storm debris, and once arriving, laid her eggs. Random changes caused by descent with modification over thousands of years eventually transformed the island creatures and the mainland creatures so much that they could no longer be considered the same species. This idea made good sense to Darwin, except for one thing. The island creatures he found weren't just randomly different from their mainland cousins, they were specially adapted for island life. The Galapagos is a collection of 18 main islands, many of which are home to tortoises. The larger islands have lots of grass and vegetation. Tortoises there grow extra heavy and have dome-like shells. Some of the smaller islands have very little grass, forcing the tortoises to feed on island cactus. The best cactus pads grow on the tops of these plants. Fortunately, tortoises on these islands are equipped with expanded front legs and saddle-like shells, allowing them to stretch their necks extra long to reach their food. It's almost as if these island creatures have been perfectly sculpted to survive within their unique environments. How did this sculpting take place? Random descent with modification alone could never do such a thing. Darwin drew upon his knowledge of selective breeding to answer this question. For thousands of years, farmers have been taking wild plants and animals and through the process of selective breeding have sculpted the original wild forms into new domestic forms much better suited for human use and consumption. The process is slow but simple. If a single plant produces 100 seeds, most will grow to be nearly identical to the parent plant. A few, however, will be slightly different. Some variations will be undesirable, smaller size, bitter taste, vulnerability to disease, and so on. Other variations will be highly valued. Thicker, sweeter leaves, for example. If a farmer only allows the best plants to reproduce and create seeds for the next crop, small positive changes will add up over multiple generations, eventually producing a dramatically superior vegetable. You might be surprised to hear that broccoli, cauliflower, kale, Brussels sprouts, and cabbage are all just different breeds of a single type of weed commonly found along the shores of the English Channel. The evolution of this original plant into all the varieties we see today was carefully guided by different farmers around the world who simply selected for different traits. It's important to note that the farmer doesn't actually create anything. Random descent with modification creates new traits. The farmer simply chooses which of those new creations are allowed to reproduce and which are not. Darwin proposed that nature itself is also capable of selection. It may not have an intelligent brain like a farmer, but nature is an extremely dangerous place in which to live. There are germs which can kill you, animals that can eat you. You could die of heat exhaustion. You could die of exposure to the cold. When parents produce a variety of offspring, nature, simply by being difficult to survive in, decides which of those variations get to live and reproduce, and which do not. Over multiple generations, creatures become more and more fit for survival and reproduction within their specific environments. Darwin called this process natural selection. Since Darwin first put forth his idea in the mid-1800s, natural selection has been studied and witnessed numerous times in nature and in the science lab. What started out as a mere idea is now officially an observable fact. Darwin's discovery has greatly expanded our understanding of the natural world. It's led to countless new breakthroughs, and it finally allowed scientists to seriously consider the idea of common descent. So to sum things up, what exactly is natural selection? Natural selection is the process by which random evolutionary changes are selected for by nature in a consistent, orderly, non-random way. Through the process of descent with modification, new traits are randomly produced. Nature then carefully decides which of those new traits to keep. Positive changes add up over multiple generations. Negative traits are quickly discarded. Through this simple, ongoing process, nature, even though it does not have a thinking mind, is capable of producing incredibly complex and beautiful creations. I'm John Perry, and that's natural selection stated clearly. That's it for this episode. If you enjoyed it, subscribe to us on YouTube and follow us on our Facebook page. If needed, I can be directly contacted from my website at statedclearly.com. Make sure to visit brain-tools.com to check out our recent video on a promising new Alzheimer's treatment. That video shows you the basics of how the brain works, how Alzheimer's destroys memory, and how a brain tools drug called MSF achieves unprecedented revival of memory.
What if I told you there were trillions of tiny bacteria all around you? It's true. Microorganisms called bacteria were some of the first life forms to appear on Earth. Though they consist of only a single cell, their total biomass is greater than that of all plants and animals combined. And they live virtually everywhere, on the ground, in the water, on your kitchen table, on your skin, even inside you. Don't reach for the panic button just yet. Although you have 10 times more bacterial cells inside you than your body has human cells, many of these bacteria are harmless or even beneficial, helping digestion and immunity. But there are a few bad apples that can cause harmful infections, from minor inconveniences to deadly epidemics. Fortunately, there are amazing medicines designed to fight bacterial infections. Synthesized from chemicals or occurring naturally in things like mold, these antibiotics kill or neutralize bacteria by interrupting cell wall synthesis or interfering with vital processes like protein synthesis, all while leaving human cells unharmed. The deployment of antibiotics over the course of the 20th century has rendered many previously dangerous diseases easily treatable. But today, more and more of our antibiotics are becoming less effective. Did something go wrong to make them stop working? The problem is not with the antibiotics, but the bacteria they were made to fight. And the reason lies in Darwin's theory of natural selection. Just like any other organisms, individual bacteria can undergo random mutations. Many of these mutations are harmful or useless, but every now and then, one comes along that gives its organism an edge in survival. And for a bacterium, a mutation making it resistant to a certain antibiotic gives quite the edge. As the non-resistant bacteria are killed off, which happens especially quickly in antibiotic-rich environments like hospitals, there is more room and resources for the resistant ones to thrive, passing along only the mutated genes that helped them do so. Reproduction isn't the only way to do this. Some can release their DNA upon death to be picked up by other bacteria, while others use a method called conjugation, connecting through pili to share their genes. Over time, the resistant genes proliferate, creating entire strains of resistant superbacteria. So how much time do we have before these superbugs take over? Well, in some bacteria, it's already happened. For instance, some strands of Staphylococcus aureus, which causes everything from skin infections to pneumonia and sepsis, have developed into MRSA, becoming resistant to beta-lactam antibiotics like penicillin, methicillin, and oxacillin. Thanks to a gene that replaces the protein beta-lactams normally target and bind to, MRSA can keep making its cell walls unimpeded. Other superbacteria like Salmonella even sometimes produce enzymes like beta-lactamase that break down antibiotic attackers before they can do any damage. And E. coli, a diverse group of bacteria that contains strains that cause diarrhea and kidney failure, can prevent the function of antibiotics like quinolones by actively booting any invaders that manage to enter the cell. But there is good news. Scientists are working to stay one step ahead of the bacteria. And although development of new antibiotics has slowed in recent years, the World Health Organization has made it a priority to develop novel treatments. Other scientists are investigating alternate solutions, such as phage therapy or using vaccines to prevent infections. Most importantly, curbing the excessive and unnecessary use of antibiotics, such as for minor infections that can resolve on their own, as well as changing medical practice to prevent hospital infections, can have a major impact by keeping more non-resistant bacteria alive as competition for resistant strains. In the war against superbacteria, de-escalation may sometimes work better than an evolutionary arms race.
So the reason that bacteria are so good at becoming resistant to antibiotics is because they have a couple ways that they can become resistant a lot quicker, a lot easier. And the first way is transformation. So transformation, if you have a bacteria that's dead, but maybe this bacteria has a DNA that's able to become resistant to certain antibiotics, the process of transformation, the DNA can be incorporated into a new bacteria cell, so they can transfer that DNA. The other way is through how living ones do it. So bacteria possess a circular DNA molecule called plasmids, and this allows them to transfer DNA between living bacteria. So if this one's good at being resistant to certain antibiotics, then it can transfer it to another one, and then the whole culture of bacteria can become resistant very, very quickly. Okay, so that's how bacteria work. We work a little bit different. So there's two types of mutations that we can have that can change our DNA. The first one's a point mutation, and that's kind of just like a typo. You switch one letter with another, right? This is similar to changing the phrase "the fox can run" to "the box can run." Yes, it changes the meaning, but you get the basic gist of it. Right, this is our original strand and original amino acids. Here I switched out a C for an A, and two of the three are still the same. Might still be okay. Not that big of a deal with a tiny little point mutation. The other type is a frame shift mutation, and this is much worse. So in the phrase, the fox can run, if I add an extra letter and keep them in 3-letter pairs, right? So I add just an A at the beginning, right? It turns into this, and all of a sudden it doesn't make any sense anymore, right? So as opposed to with the point mutation, if I add an extra letter and keep them in pairs of 3s, well, that's not good, because if I delete a letter or if I add a letter, you can see it changes, except for glycine. Glycine, it changes what everything is. So that's worse with a frame shift mutation. Okay, so here's a board question. Pause it, see if you can do these 3 questions. I'll go through it right now. Okay, what type of mutation is this? So original and repeated. It looks like a point mutation. What amino acid corresponds to the TAT in the middle there? So I look up on my chart TAT. This should be tyrosine, and then we switch it to a TAA, which is stop. So that would change things. Okay, here's another one. Pause the video, answer these questions, see if you can do it. Okay, seen a gene sequence reads that, the mutation now appears like that. What type of mutation is this? So it looks like they inserted this G, which makes it a frame shift. Okay, so the original strand, GGA, is glycine. TTA, and then GAG is glutamate. And now this switches into glycine, so the first one stayed the same. ATT, isoleucine, and then AGA, arginine. Okay, so you can see it changed it. So if a mutation is capable of being passed down to offspring, it's called a genetic disease. So cystic fibrosis comes from a mutated chromosome number 7, and people who have cystic fibrosis, they produce a lot of fluid in their lungs, things like that, things that shouldn't be happening. So when an offspring receives mutated genes, every cell in their body has a mutation, which can lead to early death in some cases. But having two sets of chromosomes can help, because if you have one parent with the mutated gene but one parent with a good set of genes, then they cannot have any symptoms, but then they might be a carrier and they could pass this down to their offspring. So if a genetic disease is caused by a recessive allele, then you need to be homozygous recessive to have it. And that's the example for cystic fibrosis. So you can see the mom and the dad are both carriers, so they don't have it because they have one good gene. They have a 25% chance of passing it on to their kids. So this kid would have it, carrier, doesn't have the gene at all. Okay. But then if the genetic disease is caused by a dominant allele, then that's bad, because that means that you can either be heterozygous or homozygous dominant to have it. The only one that wouldn't have it is homozygous recessive. So in this situation, the father has it, the mother has it, right? And any kid with a big C would have it, and this would be the only kid that doesn't have it, okay? So that would be if it was a dominant allele. And so there's common diseases that come up in diploma and test questions because they need to be caused by one specific gene. They can't be super complicated. So these are the common ones that come up. You don't have to memorize this information, but we'll talk about it. So cystic fibrosis, it talks about the mucus that builds up in the lungs, which is bad. So it's an autosomal recessive disease, okay? And about 1 in 2,500 children in Canada are born with it. Okay, Huntington disease is kind of like ALS. Over time you lose ability to move, remember events, make decisions, control emotions. And the scary part of it is that symptoms don't appear until you're 30 to 45. So usually with genetic diseases is that if it's like a fatal disease, usually people die before they're able to reproduce. But because this doesn't show up until this age, people reproduce when they're like 25. They don't realize they have it in their family or something like that. And then all of a sudden they pass the trait down and then they get it, and yeah, it's bad. So this is an autosomal dominant, which is also bad. About 1 in 10,000 Canadians has Huntington's disease. And hemophilia is different, right? These both are autosomal, but this is sex-linked and found on the X chromosome. So we'll talk about how to deal with that a little bit differently. So let's do some questions. So a couple that discover that they have a family history of cystic fibrosis. They're thinking of having a child and they go for a genetic test to be done. Both the man and the woman discover they are carriers for cystic fibrosis allele. Build a Punnett square for the cross. So they are carriers. And cystic fibrosis, let's just use R and big R because they look really different. So we said that it was a recessive allele. So this means you have it, and this means you don't, right? So for R and big R and big R, they're both carriers. They both have one of each. It's autosomal. So I'll move little r's down, big R's down, little r's across, big R's across. So what percentage probability that their child will develop cystic fibrosis? So this one will. So it's a 25% chance. What percentage probability the child will be a carrier? So they have 2 children that will be carriers. So that'd be a 50% chance. And what percentage probability will the child not inherit cystic fibrosis? Well, that's the other 25% chance. Okay, Punnett squares just like we did before. So let's do a board question. Okay, pause the video and see if you can do this, and I will go through it right now. A man is heterozygous for the dominant Huntington allele. Okay, so Huntington, we'll use H's. This means you have it. This means you don't, because it's a dominant disease. Okay, so a man is heterozygous, so little h and big H. And he has a child with a woman who does not have Huntington's. So that means that she has both small h's. Okay, so we'll bring small h down, big H down, and both small h's across. And this is what we end up getting. What percentage probability that the child will not develop Huntington disease? So you will not if you have, only if you're just like the woman. So there's a 50% chance that they won't, and 50% chance that they will. Okay. Let's do another one. Okay, so try this on your own, and yeah, pause the video, and then you can watch me go through it after. So a woman carries one of the defective recessive alleles on her X chromosomes. This one's sex-linked, right? So that means that we have to use X's and Y's. So it's hemophilia is recessive, which means you have it. And this is a good gene, means you don't. Okay, so a woman carries one defective recessive allele. So she is a carrier. It's a big R. Okay, she has a child with a man who does not possess hemophilia. So the man is XY, and he does not have it. Okay, so let's build a Punnett square. So X little r, X little r, X big R, X big R, and then X big R across, and then Y. Okay, so what percentage probability will she have a child with hemophilia? So the only one with hemophilia is this one right here. So there's a 25% chance of hemophilia. And what percentage of the females from this cross are likely to have hemophilia? None of them, right? They both have a good allele, at least one, so they're fine. And what percentage of the males? This one will, this one won't. So there's a 50-50 chance if they have a boy that will have hemophilia. Okay, so now we're going to learn about pedigree charts. So being able to roll your tongue is caused by the dominant allele, right? But if someone's able to roll their tongue, they could have the genotype RR, R little r, big R, right? Is it possible to tell who they are, which one they are? And the answer is no, you can't tell, right? So the only way to tell is to look at a family history and see if parents can roll their tongue. And if they have a child that can't, then you can figure out the parents. And if you have grandparents and you see long lines of things, then you have more information. So geneticists use pedigree charts to trace family histories. So I have a dog. Her name is Zoe. That's what she looked like when we first got her. This is her adoption photo. And she's a greyhound, and she's a former racer. So greyhound racing, you want fast dogs, breeding with fast dogs. So there's a whole database where you can look up a lot of information. So I'm going to search up, oh, there it is right there. I'm going to search up Zoe. So her racing name was AMF Onion Soup. So I'm going to search her. There she is. I updated some photos. This is her now. So we can see her pedigree. And there she is. There's her pedigree. And you can see that her parents are Flying Penske and CTW Superlegs. And then Oshkosh Racy, she's a natural, CTW Legendary. Grace Shawnee, it's got their parents as well. You can see all her racing stats, right? Her very first race, she finished first. And then she didn't do too well near the end. Oh no, that's her very last race she finished first. Her very first race. Took her quite a while to get a first place finish. And that's why we got her when she was 3, because she wasn't that great of a racer. But yeah, you can see all this information about them. We can do test mating. So we can mate her with someone from the USA. And we can test mate her with this dog, and we can sort of see what their pedigree chart would look like if we did them together, right? So we can see their pedigree charts, we can see who their parents were, and yeah. So it shows pedigree analysis here. So in your data book on page 13, it gives you a little legend. So it tells you that females are circles, males are squares, and if you're affected, you're grayed. So this person is affected, none of these people are. Mating, you'll be attached to each other like that. So these two mated, and then you can see they have 1, 2, 3 offspring, right? There isn't one connected to this, and that's because these two mated. So, and then they produced 1 offspring. Okay, so what do we know about generation 1, right? So we know that circles are, right, if we go back, circles are females, squares are males. So we know that this is a pedigree chart for cystic fibrosis. Mom has cystic fibrosis, dad doesn't. Okay. And since we know cystic fibrosis is an autosomal recessive, so this means you have it, and this means you don't, right? So if the mom has cystic fibrosis, we know her genotype. The dad is either going to be little r, big R, or 2 big R's. We're not sure. Okay. Explain why none of the children have cystic fibrosis, right? So if we're going between the 2 of these, if we did a little Punnett square for this, so the mom is like this, right, which means she passes her recessive cystic fibrosis allele along, right? If the dad was like this, then 50% of their kids would get cystic fibrosis, right? Which probably means that the dad is this, because none of the kids have it, and he will always pass down that healthy allele. So the kids will be carriers, right? If we switch this to a big R, all the kids will be carriers, but none of them will have it. And then it shows up in the next generation, so that makes sense. How many boys and how many girls are in the second generation? Well, they had 1, 2, 3 girls and 1 boy. And then the girl mated with this boy that's not part of the family. Explain why cystic fibrosis showed up in generation 3. Well, we talked about that, right? All the kids are carriers. So so this one's a carrier. Oops, little r, big R. Right? So if this person's a carrier as well, they got a 50% chance of having a kid with cystic fibrosis, right? And they actually beat the odds that 75% of their kids don't, but this kid did because they genetically have a 50-50 shot. Okay, albinism is albino, loss of pigment, skin, hair, eyes in most humans. Severe form is called oculocutaneous albinism. It's an autosomal recessive disorder. So that's really important information. Okay. So what are the genotypes for the first generation? Well, if they have it and it's recessive, right? Recessive have it, don't. So if they have it, they have to be like that, right? And the fact that some of the kids have it tell us that it's going to be like that, right? Because if this was big R, because this person doesn't have it, none of the kids would have it. So the fact that they have it, we know that they're heterozygous, right? And then we can build a Punnett square and see what the possibilities of kids are. So bring that little r down, big R down, little r across, right? So you're either a carrier or you have it. So second generation, the genotype is going to be little r. Oops, sorry. Oops. That's for the people that have it, sorry. This is little r, little r, little r, little r, little r, little r, and this is little r, big R, little r, big R. And the fact that they had a kid that has it means that they have to be heterozygous. So what are the genotypes for the first generation for the parents to determine the genotype for all the offspring? Yeah, we can figure out the genotypes for everyone. Okay, so let's do a board question. So pause it, answer these two questions, watch me go through it. So Huntington's is autosomal dominant. What are the genotypes of the parents? This means you have it, and this means you don't. So the only way not to have it is to be two little ones, right? So this one, big H, little h, or big H, and we have to look at the kids to see what's more likely. Big H, pass the dominant allele to each one of their kids. So that means every kid would have to have it. But this is a kid, this is a kid, this is a kid, this is a kid, this is a kid. So they had 1, 2, 3, 4, 5, 6 offspring and 50% chance of them have it. So it's going to be heterozygous. Okay. And then what would the genotypes for all generation 2? Well, you'd have to look at big H, little h. So move that little h across, move the big H down, move the little h across. So if you have it, you're hetero. If you don't have it, you're homozygous. Okay, these would be the two possibilities. So all the kids that don't have it would be this, oops. Just their kids, not the mate, right? And all the kids that have it would be big H, little h. Okay, that's all. So with this one, what do you notice about all the offspring who have it? This one, this one, this one is just getting passed on to boys. Why is this happening? Well, it's mostly probably an X-linked recessive gene, right? Because if the boys have it, then they're passing it on. This mom has it, which means she has both recessive alleles, right? The dad probably doesn't have it. So the dad always passes on the X, the mom passes on one of the faulty genes to the boys, so the boys end up always getting it. The girls always get one good gene from their dad. So the fact that boys are always getting it is good indication that it's sex-linked. So let's do some board questions. Tell me what you think. Is it autosomal dominant, recessive, sex-linked Y, or sex-linked X? Okay, so pause and try to figure it out. So it's boys getting it, which lean us towards this. Okay. Sex-linked X and sex-linked Y. So if we look through, every single boy offspring gets it, no matter what, right? So because they get it no matter what from their dad, and then this one here, the girl didn't pass it on to her sons, this one is probably going to be sex-linked Y, because those boys are always getting that gene from their dad, so they have to get it. So sex-linked Y would be a better answer for that one. Okay, this one, okay, we see boys and girls are getting it. The parents don't have it, which means that they're probably both heterozygous, right? If this was so it's not sex-linked boys aren't showing up. If it was a dominant one, right, both parents would have it, right, in this situation. So it's got to be an autosomal recessive, right? Usually if it skips a generation like that, you can say it's probably autosomal recessive. Okay, pause it, see if you can figure it out. Okay, this one here, boys and girls are getting it, which leads us away from sex-linked. So dominant or recessive, we're seeing it all the way through. So let's take a look if it's recessive first. So if it's recessive, that means it's like that. And that means it's like that. It's got to be like that, right? Because if this person was like this, then they'd always get that good gene and none of the kids would have it. So it would be this situation for sure. And then half the kids get it, half the kids don't. Right? Make that R go down, R go across, like that. So they'd have a 50-50 chance, which looks like it's happening. Now let's take a look if it's dominant. So if it's dominant, this is always so tricky to go back and forth between the two. So if it's dominant, this means you have it, and this means you don't. So the only way that this person doesn't have it is if they're small r, and then this person has to be heterozygous as well, because if they were big R, then that means all the kids would have it. Okay, so it looks like, again, it's going to be the same 50-50 shot. So this one's, you can't really tell. It could be dominant. It could be recessive. Yeah. Yeah, there's not that much of a way to tell. So it could be either one with this one. So what type of disorder is this? Pause it, see if you can do it. This is a tricky one. So both the parents have it, and none of the kids have it. It's not looking like sex-linked. Boys and girls can have it. So if we're talking about recessive or dominant, so let's look at dominant first. Okay, so it could be this, if the parents have it, or it could be this, right? But that's not likely to be that, because that means all the kids would have to have it, right? So if we did a Punnett square for this, right, there's a 25% chance that none of their kids have it. Okay? But let's take a look at what recessive would mean. So for recessive, this means you have it, and this means you don't. So to have it, you have to be like this, right? That's the only way. But that means that every single kid would have it, which is crazy, right? It's skipping a generation, but it has to be dominant, because the only way, it's only a 25% chance, so they just won the lottery, right? All 4 kids, 1 in 4 chance of not having it, and they all didn't have it. So the only way for this to work is if it's dominant, right? Because if it was recessive, they'd have to have both small r's to both have it, and all the kids would have to have it then as well. Okay, any questions, let me know.